Friends, welcome back in another video from Microbiology with Chagnik. So in this video we are going to start a new chapter, that is methods in molecular cloning. Okay, so if you have remembered that in the first video of recombinant DNA technology, where I have talked with you about the big picture of recombinant DNA technology, here I have mentioned that after the once the recombinant DNA is produced and it is by attaching the gene of interest with the vector. Now we need to transfer our recombinant DNA to the host, right? And we have termed the process as transformation. Okay, so this transformation process is the is all about today's lecture. Okay, so this is going to be the introductory video, and after that I am going to discuss with you about various transformation process that is popular in recombinant DNA technology one by one in separate videos. Okay, so as I have told you that there are several transformation process for transferring the recombinant DNA, that means vector plus gene of interest, into the host cell. Okay, so before choosing the transformation process, there are some things that you need to keep in mind. Okay, so the things are, whatever the transformation process you will choose for your recombinant DNA technology recombinant DNA technology work, first of all the transformation procedure must be convenient. Okay, and it does not have the ability to physically harm the vectors as well as our gene of interest, that means our recombinant DNA, along with the host itself. Okay, because because in the host cell we need to replicate our or the gene of interest. Suppose this is the host cell, this is the red marked portion inside the big red is the gene of interest along with the vector. Okay, so this is the situation once after the transformation procedure is completed. Okay, so now the viability of the host cell is important, because this vector along with the gene of interest have to replicate itself here. Okay, and now if the host cell is not viable, then how our gene of interest will replicate, right? And again we need we need some help of host cell machinery for transcription as well as translation. Okay, because sometimes we also need the expression of the gene of interest. So the viability of host cell is very important to us. Okay, and again the featured of the recombinant DNA cannot be compromised, because this is our because the recombinant DNA contains the gene of interest, and that is our things of study, or that is what we are interested about. Okay, but if due to transformation procedure the feature of recombinant DNA is changed, then we cannot get our desired product, right? That is why first of all the transformation procedure must be very convenient. A second thing is, the transformation procedure does not have the ability to harm the the vectors along with the gene of interest, that is recombinant DNA, along with our host. Okay, that three things we need to keep in mind. That means viability of the host cell, the second is our recombinant DNA should be intact, and the third one is the process must be convenient. Okay, so now after learning about this, I am going to talk about some different procedure that is used for gene transfer. Okay, the first procedure is direct gene transfer by physical method. Okay, so here electroporation, microinjection, ultrasonication are used. Plant animals electroporation are used for plant animals and bacteria. Microinjection are used for plant and animal. Ultrasonication are used for plant. I am going to talk about all of them in details in our upcoming video. So you don't need to bother about that, because this is only the introductory part. Okay, and then in case of, we can also transfer the gene by chemical method. Okay, so method here is CaCl2 heat shock. That is important. It is performed in case of when the host is bacteria yeast. Again we have liposome packaging and fusion or endocytosis, that is plant and animals. Okay, that is used in case of plant and animals. The third one is DAE, that is dextrose transfection. It is also performed in case of plant and animals. Okay, so after using physical method and chemical method, scientists have found that the huge success of recombinant DNA technology along with gene therapy. So they have decided to use some biological methods too for direct gene transfer. Some of them I have already made a video of some of them. That one are baculovirus. So baculovirus, about baculovirus based vector, I have already made a video. If you want to know about what is baculovirus based vector, then you can easily go to my YouTube channel and search about that. Okay, so baculovirus not only used as a vector, but they also can be used as a transformation procedure. Here they are going to be infect insect, so we can only use baculovirus for transformation process of organism. Okay, second is viral transduction. In that case, it will be animals. And the third most important one, agrobacterium tumefaciens mediate transformation. It is performed in case of plant. Okay, and here it is organism. It will be organisms transformed, not transfection. Okay, sorry for that. So these three things are important. So in our next video we are going to talk about how CaCl2 and heat shock are used as a transformation procedure in case of bacteria and yeast. Okay, so till then, bye bye.